adaptive immune response has two arms: natural, innate, adaptive, acquired. The difference between natural and adaptive immune response: the first line of defense, the natural immune response, is the first line of defense. It's rapid, acts within minutes. The adaptive immune response is the second line of defense, slow, acts within days or weeks. Natural immune response non-specific. The adaptive immune response is specific, discrimination, no memory in natural immune response. There is a memory in adaptive immune response. There is a memory in adaptive immune response, not improved by subsequent infection. The natural improved by subsequent infection with increased amplitude cells. The natural immune response, the component are phagocytes, cells, phagocyte, natural killer cell, mast cells. The cells are lymphocytes. Mediator in natural immune response complement. Mediator are antibodies and cytokines. Mediator in natural immune response are complement, interferon, acute phase proteins. Features of immune response: constitutive, present period to infection, the natural, induced by exposure, amplify the natural immune response, other immune mechanisms. Adaptiveness, respond to previous unseen antigens. Diversity, recognize about 10 raised to power from 10 to 18 antigenic epitopes. Adaptive immune response, the features and the characteristics: specificity, memory, adaptiveness, discrimination, and diversity. The specificity or colonial selection theory explain the specificity feature of the adaptive immune response. Antigen epitope select the cell that carry receptor with the same configuration. Here, lymphocyte, clone, and mature. This is the antigen, the mature lymphocyte. Lymphocyte clone mature in generative lymphoid organ in the absence of antigen. Clones of mature lymphocytes specific for diverse antigen enter the lymphoid tissue. Antigen specific clone are activated, selected by antigen on product, passive, ready-made, developed by exposure, natural infection, artificial immunization. Passive, ready-made, developed without exposure, natural from mother to baby, artificial. Microbial antigen vaccine or injection, days or weeks. Challenge infection and its recovery. Active immunity and passive immunity, that is the comparison between active immunity and passive immunity. Here, when we inject serum, antibodies or cells, T lymphocytes from immune animal, adaptive transfer to naive animal, challenge by infection, recovery, and immunity occur. The specificity, yes, this is specific. There is no memory. When we challenge the infection after injecting the vaccine, after days, the animal recover and the immunity developed. There is specificity and there are memory cells. Active acquired immunity, primary immune response after the first exposure, secondary immune response after the second exposure. The serum antibody titer here in the y axis and the time by weeks in the x axis. Here the primary response, activated B cells, memory cells, primary anti-X response. In secondary anti-X response, there is primary antibodies and the activated B cells. The activated B cell increase. Types of immune response: humoral and cell-mediated immune response. Humoral immune response provide protection against extracellular infections, cells, mediators. Cell-mediated immune response provide protection against intracellular infections, and there are cells and mediators. Humoral immunity and the cell-mediated immunity: the microbe, extracellular microbe, phagocytosed in macrophage. Intracellular microbes like viruses replicate within the infected cell. Responding lymphocyte, B lymphocytes in humoral immunity. Helper in cell-mediated immunity, the helper T lymphocyte and cytotoxic T lymphocytes. The transferred by serum antibody block infection or eliminate intracellular microbe. The function of the antibody in this process. Here in cell-mediated immunity, activated macrophage kill phagocytosed microbe. Cytotoxic T natural killer cell kill the cell which is containing virally infected cells or tumor cell. Cells of the adaptive immune response: antigen recognition, effector function. B lymphocyte recognize the microbe, antibody formation. B lymphocyte neutralize of microbe, phagocytosis, complement activation. Helper T cell, T lymphocyte. Here activation of the macrophage, inflammation occur. Activation, proliferation and differentiation of T and the B lymphocyte. Cytotoxic T cell, CTL. Killing of the infected cell, function killing of the infected cells. Natural killer cell, killing of the infected cell. Phases of adaptive immune response: cognitive phase, activation phase, effector phase, decline and memory. Here recognition phase and native T cell, clonal expansion, differentiation. Here the activation phase, effector lymphocyte, elimination of antigen. The humoral immunity and cell-mediated immunity develop. Decline phase, apoptosis and the memory cell formation, surviving memory cell. Here the time on the x-axis, days after antigen exposure. Cognitive phase, specific receptor on the lymphocyte recognize and bind to specific antigen. BCR or antigen receptor bind soluble forms of foreign protein and polysaccharides. T-cell receptor complex bind short peptide of protein antigen expressed with major histocompatibility class 1 and major histocompatibility class 2 on antigen presenting the cell that require also participation of co-stimulatory molecules and adhesion pairs. Here the lymphocyte, antigen receptor, microbial antigen, molecule produced or induced by microbe, lymphocyte proliferation and differentiation, the two signal requirement of the lymphocyte. Activation phase, activation of T cells. Activated T cells express secretory interleukin 2 and more growth occur. Activated cell undergo proliferation, clonal expansion of antigen specific cell and differentiation occur. Differentiation, CD4 differentiation, T helper 1, T helper 2, CD8 differentiation, cytotoxic T cell. Effector phase, the effector function cells, T helper 1 inflammatory T cells, macrophage activation, phagocytosis, T helper 2 B cells activation, formation of the antibodies, CD8 cytotoxic T cells killing the target cells, natural killer cell activated by interferon, killing of the virally infected cells. Here macrophage activation enhanced microbial killing. Interferon gamma. Here the naive CD4 proliferate and differentiate to T helper cell by interferon gamma and tumor necrosis factor. The neutrophil activation enhanced microbial killing, and the complement binding and opsonization antibodies and the macrophage activation enhanced microbial killing. The B cells differentiated, produce antigen, mast cell degranulation, production of neutralizing antibodies, human IgG4, mouse IgG1, the activated macrophage, suppression of the macrophage activation, eosinophil in the parasitic infection, target cells CD4, granulozymes. Granulozyme enter through perforin holes, activation of the caspase. Here apoptosis of the target cells is the end product. Activation of B cell, binding of antigen to BCR, deliver the antigen inside the cell, digestion of the antigen into peptides, expression of its surface with major histocompatibility class 2, recognition by T helper 2, secretion of cytokines, B cell proliferation, differentiation, effector cell, phase cell, the recognition phase and the activation phase, helper T cell other stimuli. Here the resting IgM, IgD mature B cells, the antigen, activated B cells, clonal expansion, IgG expression on B cells, high affinity IgB expressing B cells, the effector phase or hormonal immune response, antibodies, naturalization of the microbe and toxin, opsonization and phagocytosis of microbe, antibody-dependent cellular cytotoxicity, lysis of the microbe and phagocytosis of the microbe, opsonizing with complement fragment, and the inflammation, effector phase, isotype which plasma cell transfer to antibody production, early IgM, later IgG, IgE. This process is regulated by T helper 2 cytotoxins such as interleukin 4 and tumor necrosis factor. Why immune response are self-limited? The body is always in balanced state called homeostasis. When the induced antigen is eliminated, no survival stimulus, no activation. Lymphocytes perform their function for limited time, switch off, feedback regulation. Factors that regulate immune response: genetic control, cellular control, molecular control, tolerance. Regulation of immune response. The genetic control. Our genes play a major role in determining whether we will make poor response to various antigens, good response, average response. T helper 1 cytotoxic, regulation of immune response, the cellular control or cytokines, their interaction between T helper 1 and T helper 2, interferon and interleukin 2. T helper 2 secrete cytotoxins, interleukin 4, interleukin 10. The interaction between both of them, induction of cells reactive to antigen in positive way. A positive cell in turn stimulate a negative way. Suppressor cell, helper cell. Inhibitory effect of suppressor cells, production of suppressor factor, production of inhibitory cytokines. interleukin 10, interleukin 13, absorb the necessary growth factors such as interleukin 2, antigen recognition, T cell proliferation, effector function of T cells, contact dependent inhibition of T cell response, cytokine mediated inhibition of T cell response, regulatory T cells. Here the antigen presenting cell and the interleukin 12, native cells, effector T cells, activated macrophage, interferon gamma, cytokine mediated inhibition of T cell response, regulation of immune response, cellular control, apoptosis. Apoptosis is a mechanism by which the effector cells could be deleted after eliminating of antigen. Surviving cell express the anti-apoptotic protein BCL2, antigen recognition, T cell response, apoptosis, activated T cells, expression of Fas and Fas ligand, and the apoptosis. Fas mediated activation induced death of T lymphocyte, suppression of B cells. Two ways of B cell suppression: T helper secrete interferon, affect the macrophage suppression of the B cell. Antibody feedback inhibition of B cell, simultaneous interaction of antigenic portion of complex with membrane antigen or antibody complex with Fc receptor on the same cell, block antibody production, block in the B cell receptor signaling, and this is a process. Antigen antibody complex bind to the B cell Ig and Fc receptor associated remove the phosphate in B cell receptor complex. Molecular control, T cell antigen presenting cell interaction through CD28 B7 co-stimulatory molecules, mechanisms of helper T cell mediated B cell activation, antigen presenting to helper T cell activation of the T cell expression, CD40 ligand cytokine secretion, activation of B cell by cytokine and CD4 ligand, ligation, B cell proliferation and differentiation, mechanism of helper T cell mediated B cell activation, tolerance regulation in the development of competent cells by tolerance to self-antigen breakdown, autoimmune disease, regulation of the mature competent lymphocyte to antigen breakdown, autoimmune diseases occur. Factors that determine immunogenicity, tolerogenicity of antigen. These factors: the amount, the persistence, the portal of entry, presence of adjuvant, properties of antigen presenting cell, fever, stimulation of immune response, the amount, optimal doses that vary from different antigen, fever tolerance, high doses, persistent, short-lived, eliminated by immune response, prolonged, repeated T-cell stimulation induced apoptosis, portal of entry, subcut, intradermal, intravenous or oral, presence of adjuvant. The antigen with adjuvant stimulate helper T cell, antigen without adjuvant tolerogenic, properties of antigen presenting cell, high level of co-stimulators, low level of co-stimulators and cytokines, newly emerged immature clone of lymphocyte, maturation of the clone not specific for self-antigen presenting in generative organ, central tolerance, deletion of lymphocyte specific for self-antigen present in generative organ, immune response to foreign antibodies, proliferate tolerance, deletion or energy of lymphocyte that recognize self-antigen in peripheral tissue.